our first one of the 2018-2019 academic school year. I would like to give a welcome to Dr. Den Hollander. Dr. Hollander was originally trained as a molecular biologist. Early during his PhD, he was inspired by the rapid expansion of data generation and advances in computational biology. He acquired a PhD in bioinformatics at the Medical Statistics and Bioinformatics Department of the Leiden University Medical Center. While doing so, he elucidated the epigenetic and transcriptomic profiles of artificial cartilage using high-dimensional data combined with unsupervised machine learning techniques. Hereafter, he joined the toxicology department of Leiden University as a postdoctoral researcher, utilizing microscopy data in order to predict potential idiosyncratic drug-induced liver injury due pre-market drugs. Currently, he is employed as a research data analyst at the Trimbos Institute, where he aims to bring innovative machine learning techniques to a field previously dominated by conventional statistics. So Dr. Den Hollander, welcome. Thank you so much for joining us today. I will turn the floor over to you, and I would just like to let everybody know before we get started, feel free to ask questions throughout the presentation. For those of you who are joining from afar, you will have to unmute your microphone. We mute everyone on our end to decrease feedback. So if you have a question, feel free to unmute yourself to ask your question, but then make sure you remute yourself. We are also recording the seminar and we'll make it available after the presentation. Thank you.
All right. Thank you very much for the kind introduction. Can everybody hear me well?
Yes, we can hear you great on our end.
All right, that's good. So yeah, so I'll be talking about some unsupervised machine learning techniques that I acquired during my PhD and now apply it to a more social field, and in this case specifically the Scottish Wellbeing Study. Some of the techniques I'll be showing you are probably familiar, but I hope to give it a little twist on how you could visualize these kinds of results from these analyses to basically give you new insight in your data. And I guess I will also be introducing some novel techniques. But I have tried to keep the mathematics and the technical parts, I dialed it down so that the concept should be clear. And I hope comprehensible for everyone. So, first off, let me see if it works. It does. So machine learning. So basically within machine learning, there's two major branches of machine learning called supervised machine learning. And I guess most people are familiar with these kinds of techniques. They work with known labels and they test explicit hypotheses, such as linear models and regression trees. Having known labels and explicit hypotheses means as much as that you know a priori which models you want to fit, which variables you want to check in relation to the outcome you're interested in. And at the other side of the spectrum, you have unsupervised machine learning. Now, unsupervised machine learning handles unknown labels and is more of an exploratory nature. Unknown labels means as much that, of course, to you as a researcher, the labels of your samples are known to you, but you don't tell it to your computer, basically. So you ask your computer to run these algorithms without knowing which variables you're interested in, and let basically the computer sort it out where there's patterns in the data. There's a whole array of these techniques, and one of them is principal component analysis, which is probably familiar to some of you. And the other one is hierarchical clustering. And both of these techniques I will be showing to you within the Scottish Wellbeing Study, and at the end combine the two to get some innovative visualization of your data, which hopefully leads to generation of hypotheses as opposed to specifically tackling them. So you would apply these kinds of analyses for really upfront, early in your study, to get to know what variables are related to one another and to what outcomes. So for those who are not that familiar with principal component analyses, what you basically do is if you have, for example, a number of samples, which are here plotted in blue, and you have two variables, in this case, the x-axis and the y-axis, and you will be interested in, is there a line I can draw through these points that cover the most variation? Because that will be probably more informative than just the x-axis and just the y-axis. So principal component analysis does that for you. So it basically draws a line through your cloud of data and then iteratively moves that line until it finds a line that is least, that is covering the most variation. So what you basically done here is you've reduced your two-dimensional data set to a one-dimensional data set along this principal component axis. Now this can be very useful to do. And you can do it in an arbitrary number of dimensions. So here I've reduced two variables, x and y, to a single variable, the first principal component, but you can imagine that if you have a third variable, which is just the third dimension axis, you could also draw a line through this cloud of data points that, and then find this axis of most variation. Now, why is this useful? One of the things you could immediately do with that is that if you have a data set where you want to know whether there's outliers in your data or weird relations that you would not have expected, you could visualize it using this. So here on the left side here, on the left graph, you have the first principal component on the x-axis and the second principal component on the y-axis. If you plot these out to one another and you plot your samples in there, you can immediately see that there's a clustering of certain samples here. I've colored them by, well, in this case, donor, it doesn't really matter. But for some reason, there's this weird green dot all the way at the right, which is clearly an outlier in your data. Removing that data point, rerunning your principal component analysis, and plotting the first and second principal component to one another, you see that you've now removed this outlier. So you could also use it as a basic quality control of your data. Why is this interesting for the Scottish Wellbeing Study? So the Scottish Wellbeing Study is a representative population-based cohort documenting mental health, and there's a specific focus on suicide ideation and attempts. There's almost 3,500 people in there, about 50% is female, and the mean age is 25. Within the study, there's over 180 questions were asked, covering the mental health of these people, such as current suicidal ideation, depressive symptoms, stress, impulsivity, acquired capability, all kinds of personality traits, and then also some demographic points and outcome measures. Now, applying principal component analysis to this data set helps in getting to know the data without a priori tackling a hypothesis. So if we do this principal component trick on this entire data set, and we look at the first 5 principal components that I've drawn out of these 183 variables that have been measured, and color these now by outcome measures that are of interest to you. For example, age. You can see here that if I plot the principal component, so on the x-axis there's the principal component 1, 2, 3, 4, and 5, and on the y-axis you also have these 5 components, and you could hereby visualize whether there's clusters in your data. Now, from it, in this case, light blue is young age, dark blue is older age. And from this picture, you could see that there's no obvious clustering by age, which is nice, because if you see an age batch effect in your data, you might have to do some pre-processing before you continue with your data. Now, but if I then color by sex, you see that there's basically no clustering happening. But if I looked at the 5th principal component all the way at the right, the most right column, or the bottom row, you see that there's a suggestive separation of the sexes. And if you then check the correlation coefficient between your in this case, the 5th principal component and sex, you see that there's a 0.15 correlation there. So this is just a very basic way to visualize potential batch effects in your data.
Dr. Den Hollander, may I ask a question if you wouldn't mind going back to your previous slide, this one, yes. Can I ask, so if I am following correctly, you've employed a variety of scales and all of the questions in the scales are 183, total of 183 questions?
Yeah.
Yes, and so you've identified 5 principal components. I am curious to what extent these empirical results match in any way I don't know, maybe you have 10, 15 scales. Is there any kind of clustering that confirm the theoretical structure of those scales?
I'm not really sure what you're meaning by scales. So could you?
I'm going to try to work on the vocabulary because it depends on the field. The instruments, the instrumentation that you've used. If you go on a couple of slides up, there was a depression scale and...
yes. So these are all 183
yes.
Variables scaled to between 0 and 1. So
yes.
Yeah.
And the 5 principal components, the questions that fit into the 5 principal components, is there any kind of let's say common thread within each principal component? So do the depression items go in the same?
You're going too fast. So
sorry. Okay.
No, it's okay. But it's good that apparently the flow of the presentation is well.
No, it's great.
Your question will be answered in, I guess, 1 or 2 slides. So.
All right. I'm holding on.
So I've taken the first 5 principal components here somewhat arbitrary. You could also take the first 100, of course, or the first 3, whatever. But for visualization purposes, this is I took the first 5. So. And the question is indeed, so what do these principal components actually reflect from your data? So if I take a look at the first principal component, I see that there's a very strong negative correlation with the first principal component and in this case, general well-being, as you can see at the top here. So for the first principal component, you see that in general, there's more light blue dots at the bottom than there is at the top. And this also goes for optimism, which is what you would expect, because general well-being and optimism generally, well, you would expect a correlation between the two. Now, what we also see is that there's a positive correlation with stress, which is also to be expected given that it's a negatively correlated with general well-being and optimism. So the first principal component seems to reflect general well-being, how good do people feel overall. If we then go to the other ones, we see that the second principal component, so this one appears to be positively correlated to careful thinking, which is one of the part of the questionnaire that were asked. Impulsivity is related to the third principal component. And then we have the fourth and the fifth, which is perfection. And apparently love from others is fairly strongly correlated with the fifth principal component. Does this answer your question?
Yes, thank you.
Okay. So far there's not much surprising, but it's a fairly easy way to show you what's happening with your data without diving into technical details. Now, there's a way to visualize these relations, of course, the individual items to these principal components. So if I look at well-being, which was strongly correlated to the first principal component negatively, we indeed see that there's a fairly strong fit between well-being and the first principal component. The other way around, if you look at again at the first principal component, but now plot stress at the x-axis, we see that there is a positive correlation. For binary outcomes, visualization is done a bit differently. So here I plotted the first 5 principal components, the distribution along the first 5 principal components, and colored them by sex. So here in green we have males and in red we have females, and how they score on the different principal components. And as we saw earlier, there's some suggestive separation of the sexes at the fifth principal component, which also nicely shows here at the most right graph. Now, there was the main focus of the study was on suicide ideation. So instead of just looking at sex or other demographic variables, it's of course interesting to look at this at suicide ideation. And here we see that there's a fairly strong differential distribution of suicidal ideation and at the first principle component, meaning that this general well-being is seems to be associated with suicide ideation, which what you would expect. Yeah, luckily I see some people who are from the 90s or earlier, because I want to transition here to hierarchical clustering. Is this familiar to anyone, this picture?
Yeah,
Okay, so there's no Monty Python fans attending. That's too bad. Okay.
Python.
Okay, yeah. So this is John Cleese basically transitioning from one subject straight to the other without a proper, well, flow. So hierarchical clustering. Is anyone familiar with hierarchical clustering? I guess not. Okay, so it's fairly easy. If you have a single measurement, so let's say depression, the distance or the difference between samples, between participants is fairly easy calculated. You just take the score of your X sample and your Y sample and take the absolute difference. That's basically a description of how different your two samples are. Now, you could also do this with multiple measurements. A third one, and now this might look a bit daunting, but this is basically just calculating a triangle, the length of is the hypotenuse? I think so. The distance between 2 points. You could do this for for 3 dimensions, and you could just generalize this across n measurements. Measurements should be here instead of samples. So this might be a little bit daunting, but if you visualize it it can be visualized fairly easily. So a single measurement is just a difference between your two samples. If you do that in a 2-dimensional space, meaning you have 2 measurements instead of a single measurement for 2 persons, you just have a triangle and you calculate the distance between these two. And this can be done for higher dimensions as well. So a third measurement would again imply a third axis, and you could just calculate the distance between samples within this hyperplane. Now, how would this work? So if let's say I have 5 samples in this case, 6 samples, and I have 2 measurements, X and Y. I want to visualize all the distances from all points to all the other points. You could do this using a dendrogram, which look like this. So this basically means that sample A and sample B are most closely together. If you in this space of the X and Y, and then further down the line, you see that D is also is furthest away, and then F and then C and E are also somewhat close together. So using this dendrogram, you basically summarized your 2-dimensional plot to a 1-dimensional plot showing the absolute distances between different points. And this trick we could also apply on our data set. So if we do that on our participants and we take into account all the 183 measure points, and we look at the distances between all of our samples, of all of our participants in this 183 dimensional space, we get a clustering like so, the upper one. And we could also do that using the individual items. So this is basically visualizing how alike the participants are and how alike the items are. Now, this is not so, not that informational, but we could use this clustering to generate a so-called heat map. So if we plot all the data as such, we have our upper participant dendrogram at the top and our item dendrogram at the left. And we now just color in the intersections between these lines, yellow meaning there's a low score and red meaning there's a high score. So immediately you could see that if you have these 3,800 samples and our 183 questions, and we space them out accordingly how alike they are, you could see that there's a clear clustering of both participants and questions. So one of the things that immediately pops out is this one. So we see here that there's this cluster of patients all the way at the left that appear to score fairly high for the green questions here and fairly low for the blue questions. And if we then take a further look at what these green and blue questions are, we see that, is this readable for you?
Yes.
Okay. We see that the blue questions are mostly items which assess positive emotions, so whether people are optimistic, whether they're cheerful, whether they experience love and emotion, whereas the green ones here are mainly negative. Negative items that are assessed, as failure, burden, pessimism. So immediately you get a sense that there's a cluster of patients or participants who score fairly low on these optimism items and fairly low on these negative emotion items. So you want to, but this is fairly qualitative. So you want to have a quantitative way to define these clusters. And in this study, we were not so much interested in clustering the individual items, because that's known for a lot of people that, of course, well, positive emotions will be correlated to one another, negative emotions will be also correlated to one another. But it would be interesting if you could define clusters of patients which are unbiasedly designated as, for example, depressed or optimistic. So we could do that using, again, going back to our dendrogram, we could just cut our dendrogram across in a horizontal way. For example, if we cut our dendrogram in this case at height 6, we get two clusters. One cluster will be AB and the other cluster will be DEF, CDEF. So this would mean that apparently A and B are more alike to one another than the other cluster of samples. And you could, of course, differ in your decision where to cut the dendrogram, yielding smaller clusters of participants. But you want to have some sort of a quantitative way, unbiased way to decide where to draw the line and thereby generate these patient profiles or patient clusters. You could do that using a technique called WGCNA, which is a technique often used in gene expression analyses, basically to look for genes which, for whatever reason, behave the same way across different situations. So we have basically two parameters that we have to set up front. So we want to have a minimal module size. So that means how many patients do I want to have, participants, do I want to have at least in each module? And you want to you need to define the minimum distance between these modules, which basically means how far are these modules from one another until we decide, no, these are fairly close, so we want to have them to be in a single module. So you get these clusters of modules or clusters of patients, which now you can get the first principal component from. And the next step is then, if you've generated all of your clusters, and then you individually check all participants to what extent they relate to those modules, the first principal component, and if they are in a module but more strongly correlate to another module of the first principal component, you move that person to that module. So if we now again have our dendrogram at the top here, it looks a bit differently. These are participant dendrogram, and by using these two parameters, we can and then iteratively relocate these participants to the modules they are most strongly correlate to. We get all these modules defined by color at the bottom here. Now, this doesn't look very appealing, but this should help. So here we have all of our participant modules sized by the number of participants in there. So we have a large gray one, which apparently contains a lot of participants that are, for whatever reason, very alike. And we've got a number of smaller modules containing other patients, which are, for whatever reason, within these 183 items look like one another.
May I ask a question?
Sure.
This is Camelia again. I just want to say it back to you in lay language to see if I understood.
Okay.
So what you're doing with this modeling is determine which patients most strongly resemble each other by their answers to specific items.
Yes.
And so, for example, you have a large number of patients or participants in the large gray circle that are strongly related to each other through mostly their answers to those items towards the bottom of the diagram. Am I interpreting it correctly?
Yes. So if we...
Yes. And then what I don't understand then very much, and I would love to, is your point number 3. And I don't know what it means to iteratively relocate participants.
Okay. So if you have this dendrogram here with these modules plotted, so these circular, not the modules, this is done at step 1. So their participants are located into a module based on the 2 parameters I gave, so the distance and the size you want to have your modules to be, well, the minimum size. And that's step 1. Then you will be checking for each participant, do they resemble the first principal component of the module they're in best, or is it another module? If it is another module, so not the one it's originally placed in, then you relocate that person to the module it's strongly most correlated.
I understand. Thank you.
Then the second step is calculate again all the first principal components from these modules, check whether participants are in the module they most strongly resemble. If not, move it to the one it does, and then do it again, do it again, do it again.
Yes, until you minimize, well, I'm not sure exactly what the terminology would be, but you minimize some leftover variance or...
Exactly. Yeah, exactly. Yeah, that's exactly what's happening. Yeah.
Thank you, Dr. Lindholm.
Okay, so to recap, because it might be a little bit daunting. So we have this data set of 3,480 participants, 183 items. Using hierarchical clustering, we cluster them into these modules. And then next, in using principal component analysis, we iteratively relocate those participants to the modules they mostly resemble. One further step is that if you now cluster these modules, which you just saw, we have this entire analysis pipeline basically, which is called WGCNA. This is often used in molecular biology. Yes, so turning the axis now into a circular one, it gets somewhat more visually appealing and easier to comprehend. So this is the same module dendrogram as I just showed you before, only here it's circularized. So we're gonna get our gray large module at the top and all the other modules surrounding it. Now we have our produced data set, which we can use now to visualize outcomes of interest. So for example, if we now take a look at suicide ideation and we plot the suicide ideation scores of these participant modules on top of the map, from blue ranging to red, blue will be below the average, red will be above average. We could easily identify 4 modules here, which for some reason have a very high score on suicide ideation. Now we can take a look at other variables, such as one of the items it showed differences between these 4 modules. So for these 4 modules score high in suicide ideation, but are apparently located at a different position in the dendrogram. So they do behave differently. And one of the things that they behave differently on is being a careful thinker. So if we look at the modules which are in red at the suicide ideation map, and then look at those modules in the careful thinking map, we see that, I'm gonna skip through because I need the numbers of my modules here. Here we go, yeah. So if you now take a look at module 3, for example, in the careful thinker map, we see it's blue. So it's scoring lower on careful thinking, while the other 3 suicide ideation modules score high. If we take a look at the item, whether people feel that people like them, we see it the other way around. For module 3, they score on basically average on whether they think people like them, but the other 3 modules score lower on this item. We can take a look at sadness. Sadness is fairly highly correlated with suicide ideation itself, which you might also expect for guilty feelings, but here you see that there's some difference between module 3 and 4. So this could give us some insight into these people who, for some reason, have the suicide ideation, but might have a different, different factors contributing to that.
Dr. Von Hollander,
Yes.
can I ask a question just to clarify my mind? So if I am not mistaken, these 5 are your 5 principal components. Is that correct?
No. So what do you mean by these 5? These 5 graphs, you mean?
Ideation, careful thinker, people like me, or are these original, sorry to call them scales, assessments?
No, it's fine. Yeah, so these were not among the 183 items, but these were outcome measures and alongside a number of demographic measures, scales. So the data that is used to color in the modules here is not the data used to actually cluster the patients.
Okay.
So you're basically superimposing other measures than you've used to generate these modules, and then see whether the data that was used to generate these modules has a relation to certain outcome measures.
Oh, that's great. Thank you.
Because what you're suggesting would also work, but you would expect fairly strong correlations, because those are the items used to actually make the modules. So you'd rather not use those.
Yes. So these are additional measures.
Exactly. Yes, and of course they're related.
Yes.
Well, that's obviously clear from the graphs here.
Yes. Thank you.
So we could further dissect these different measures to suicide ideation. So if you now take a look at our guilty feelings map, which is the same as we saw before, we could start diving into the differences between these 4 modules. So we have here module 1, 2, 3, and 4, and then the guilty feelings scale was from 0 to 3, basically meaning no, seldomly having guilty feelings, and 3 meaning a lot of guilty feelings. And we see that for module 4, there's a large number of people which score fairly high on the guilty feelings measurement, while the other 3 score somewhat lower. So this apparently means that although these people are having suicide ideations as the other clusters, they are apparently more strongly driven by guilty feelings than the others. We could also do this for sadness. So here there's a difference. Let me go back one. So here we see that module 4 is scoring higher on guilty feelings compared to module 3. But if you take a look at sadness, we see this is the other way around. Again, visualizing this using these pie charts, we see that module 3 is scoring higher on average, because there's only so many people scoring 0 on this measurement. So going through all these extra outcome measures, we see we can start looking, okay, to what extent are these modules behaving differently across the measurements. So sadness and guilty feelings we already had. If you take a look at easy to relax, we see that they all score, should be the other way around, fairly low. But if we now take a look at modules at another position in the dendrogram that are also scoring high on these measurements, which appear to be coinciding with suicide ideation, we see that they're behaving quite similar across these items, but for whatever reason they're not having suicide ideation. So one of the questions you could have is, what does these modules make different? These modules that are scoring high on these negative aspects, but not on suicide ideation. So what's different between these people? This is not a question I have an answer to, but these dendrograms allow you to mine your data in that sense, and to come up with questions like these, which might not be obvious from just handling your data. What's obviously more interesting is whether these modules are able to predict certain events in the future. So the data set also has a 2-year follow-up. And if we now take again a look at these guilty feelings and sadness modules colored by guilty feelings and sadness. And we also take a look at suicide ideation 2 years down the line. We see that it coincides with sadness, but it does not with guilty feelings. So guilty feelings and having suicide ideation doesn't seem to be a stable correlation across time, while addressing sadness at time point 0 appears to be predictive, or at least correlated with having suicide ideation 2 years later. Now, these are fairly straightforward questions you could ask, which the original researchers setting up the data set were wanting to ask, but there's also other fun stuff we could do. So what I just actually noticed this morning is that there's a cluster who appears to be drinking lager more often than other people do. And so there's also a cluster which rather drinks other beers. There's a cluster which apparently likes wine. Now, this is, of course, well, somewhat funny, but if you now take a look at a specific question that is asked, if I have to stop pursuing an important goal in my life, it's easy for me to stop thinking about the goal and let go. And if you now take a look at our lager, other beer, and wine modules, we see that the other beer module scores fairly low, the wine module scores fairly high, and the, I have to say it correctly, so wine scores lower, other beer scores lower, and lager scores on average, telling me that apparently drinking lager appears to help forgetting unattainable goals, whereas drinking other beers or wine does not. So this is a bit of a joke, of course, but the nice thing is that if you have generated these graphs, you could easily mine your data intuitively and visualize what's going on, and might help you come up with research questions or specific hypotheses which you might not have thought of before. With that, I would like to end. Thank Professor Rory O'Connor, who generated the data set, and the Prosper Consortium, which I'm collaborating with on these efforts. So if you have any other questions, shoot.
Great, thank you so much for that presentation. Maybe around.
Thanks.
I actually do have a question. One of the things that I think I really appreciate about kind of population-based surveys is the potential for understanding trends and then using that as a leverage point for interventions or prevention research. So I guess I'm wondering, has that been something that you've been thinking about, or how can these techniques that you're using be applied for creating prevention efforts or evidence-based interventions?
Yes, so that's a very good question. That's exactly one of the things we're addressing right now. So if we are able to predict, in this case, suicide ideation 2 years later, maybe even further down the line, we might be able to identify participants that require some other type of intervention than other clusters of patients do, because they behave the same, but they behave differently when compared to other patients in other modules. And the nice thing about this unsupervised way is that if you specifically will be looking for participant profiles that correlate to longitudinal outcomes and compare it to those who do not correlate with that, then you're already imposing your theoretical framework on the data. But if you do it like this, and the data is showing you a separation between participant modules based on this longitudinal outcome measure, there's no need for superimposing a priori knowledge. It's just the data telling you these items are, for whatever reason, correlated to one another without assuming anything upfront. So we're generating profiles with the goal of predicting whether people need certain kinds of interventions, and will be even more nice to have to be able to ask the specific questions to new participants and locate them into a module for which we then know whether they are more likely to maintain their suicidal ideation. Does that answer your question?
Oh yeah, that's great, thank you.
I have a question that is very pragmatic. Can we get your code?
Well, sure, that's fine. I'm still working on it, so it will be. I'm trying to build in such a way that data naive scientists can work with it, so it's really easy access.
So at this time, I don't think we have our expertise in the lab. I have a minimum exposure to R.
Bob and Teresa.
Bob, so we do.
No R.
Okay, yeah. So we have some expertise in the lab. So we would be grateful to try perhaps some of this work on our own assets.
I will probably be building a website implementation of this, so you could easily upload or do it locally, so you're not actually uploading your data to a different server, which is running on your own computer. But all you have to do is basically load in your data set and then run the algorithm through a local running website. And so you're able to get these visualizations going as well. So sure, yeah, I'll be showing the code.
That's amazing. Thank you.
Can this, another question I have, because this technique is exploratory, I'm wondering when you find something in your data that's maybe disconfirming, right? It's something that you maybe haven't seen in the literature. What is the potential for these techniques to then improve these types of surveys? Because usually what I would expect is that surveys are usually created with some type of theoretical grounding, or you've gone into the literature and so you kind of know the questions that you want to ask. But with your approach, what you're finding is maybe we maybe haven't asked all the right questions. So how can this technique be used to maybe improve future population-based surveys?
Yeah, so I think you put as we would say in the Netherlands, put the finger on the sore spot. So this technique is obviously hindered by if you offer your participants a fairly narrow set of questions asking on a fairly narrow topic, it is hard to differentiate between these. So you'd rather have a data set with a large array of different kind of questionnaires. So you could, along different dimensions, see whether your patients are participants are like. Now, if you now see patterns come up which you would not have expected, it could usually mean two things. So, or one, it is something really interesting. So you might want to dive into that. Why do we see this? Well, it was unexpected. What specific items cause this correlation to exist? Or it could be a technical artifact in that sense. So you will be doing these kind of analyses really early in the research, and if you then find these kinds of patterns, you have to dissect why you would find these unexpected patterns. If it does not appear to be a technical artifact, then you might want to ask different or other kind of questions in the future.
Mm-hmm. Great, thank you. Any other questions? Or anyone online?
I hope it wasn't too technical.
No, it was great.
All right.
The one thing I'm thinking about is sample size requirements, but that obviously would depend on the number of items that you're able to put into the model.
Yes, so preferably you would have both have a large sample size and a large number of items, which are, well, they can be somewhat related, but rather as diverse as possible.
And in your particular case, Dr. Den Hollander, your sample size was about 20 times larger than the number of items. Is there sort of a rule of thumb about the relationship, the ratio between the items and the number of participants?
Not that I'm aware of. It also depends on the distribution of your specific items or skills, whatever you want to determine. So not that I'm aware of. I think it depends on a lot of things.
Thank you.
Any other questions? No?
All right.
Well, thank you so much. I think I found this to be fascinating, and I'm very excited, especially for your website to come up, because I know we'll all be visiting it. And we really haven't gotten into, I think, a lot of data visualization in our lab, and I know that's something that we would like to get into, especially as we've been scaling up our interventions and kind of reaching these larger sample sizes. So getting into different ways to visualize the data, I think, is something that we're especially interested in. So thank you so much.
All right, I'll let you know once the website's up and running.
Great, thank you.
Thank
All
you.
right.
All right, take
Bye-bye.
care.
Thanks, take care.
Bye.